Hello students, let us start with the next topic, that is megasporogenesis. So what is megasporogenesis? It is development of megaspore from megaspore mother cell by meiosis. Remember, this is gametogenesis. So of course the type of division will be meiosis, a reduction division. So megaspore is produced at a micropylar region which is surrounded by nucellus, and this megaspore is a large cell with dense cytoplasm and prominent nucleus. So do write this definition. What is megasporogenesis? Megasporogenesis is the development of formation of megaspore from megaspore mother cell. Now this kind of development is known as monosporic development. What is monosporic development? It is when only one functional megaspore develops into the female gametophyte. That is, the method of embryo sac formation from a single megaspore is termed as monosporic development. We will study in detail why am I saying that the embryo sac has developed from a single monospore, because when megaspore mother cell undergoes meiosis, it will produce 4 megaspore. Again, meiosis will produce number of cells 4, but type of ploidy, that is the number of chromosome, will be n. So 2n is now reduced to n. So megaspore mother cell produces 4 megaspore, out of which 3 degenerate and only 1 remains functional, and this one functional megaspore develops into embryo sac or female gametophyte. Because the embryo sac has developed from a single megaspore, it is known as monosporic development. So I want you to write these two definitions, what is megasporogenesis and why or what is monosporic development. It is the development of embryo sac from a single megaspore. Okay, there are few questions that you need to take care of, like why does megaspore mother cell undergo meiosis? See, we needed only one functional cell to produce embryo sac. Then why did we produce 4? It is because the meiosis is compulsory to maintain polyploidy of the gametes. Megaspore mother cell had 2n number of chromosome. After meiosis, the embryo sac that is formed has got n number of chromosome. Another question that can be asked is, why do 3 megaspore degenerate out of 4? So see, if they don't degenerate, what will happen? 4 embryo sac will be formed. After fertilization, 4 embryos will be formed. So there would be multiple embryo at the same time, and all of them may not survive. So it is better to have just one embryo. When you talk about ploidy of the different structures associated, nucellus and megaspore mother cells have 2n number of chromosome, functional megaspore, female gametophyte and embryo sac have n number of chromosomes. Now, let us talk about embryo sac in detail. What happens to the functional megaspore to form embryo sac? Now, nucleus of functional megaspore, it divides mitotically. Remember, gametes are formed, meiosis is not required anymore. Now, all the divisions will be mitosis. So, functional megaspore divides mitotically. So, one nucleus will give rise to 2 nucleus. They move to opposite poles. Excuse me. Now the embryo sac is in a 2 nucleate state. It again undergoes mitosis, produces 4 nucleus. Again undergoes mitosis, produces 8 nucleus. Remember, all these divisions are free nuclear divisions. What do you mean by free nuclear divisions? It is the nucleus is divided, but it is not followed by immediate cell wall formation. So now what you have is a 8 nucleate stage. Out of this 8 nucleus, now the cell walls will be laid down, and out of the 8 nucleus, 6 are surrounded by cell wall and they are organized into different kind of cells. 2 polar nuclei will not develop cell wall, they will remain below egg apparatus in one central cell. So now the cell walls will be laid down to give you a typical female gametophyte or embryo sac. So far we have 8 nucleus, 6 of them will form cells, 2 remain without cell wall. Let us see what kind of cells. So distribution of cells goes like this. 3 cells at the micropylar end form egg apparatus, out of which 2 are synergid and 1 is egg cell. The cellular thickenings at the synergids near the micropylar tip will form filiform apparatus. What is this filiform apparatus? Its function is to guide pollen tubes into synergid. So that is about 3 cells at the micropylar end. Now coming to the 3 cells at the chelazal end. They will form antipodal cell, and one large central cell will have 2 polar nuclei. So what kind of structure do we have now? We have a 8 nucleate and 7 cell structure. Let me show you the same thing in the form of diagram that is given in your textbook. So this is your cell where you have a micropylar end, a nucellus, and inside you have megaspore mother cell. This megaspore mother cell divides to give rise to a dyad. It again divides mitotically to give rise to a tetrad, and out of this tetrad 3 degenerate, 1 remains. Now it undergoes mitosis again, where 1 nucleus divides to 2. It migrates to the poles. 2 further divide to give you 4 nucleus. 4 nucleus further divide to give 8 nucleus. Out of them, 3 remain at the micropylar end, 3 remain at the chelazal end, and 2 come in the center. So you have 2 polar nuclei, you have 1 big central cell, you have got 2 synergids and 1 egg cell, and here you have got 3 antipodal cell. अब क्या करेंगे? अब इसको उल्टा करेंगे, that is inverting it. Micropylar end comes here and chelazal end goes here. So this is your final embryo sac, where what do you have? You have 3 antipodal cells at the chelazal end, 2 synergids at the micropylar end, and 1 egg cell at the micropylar end. So total number of cell will be 3 plus 3 6, and this one big large central cell. So total 7 cells. What is the total number of nucleus? 3 plus 3 6 plus 2 8. And what is this filiform apparatus? Filiform apparatus is the cellular thickening at the micropylar end of synergid which guides the pollen tube into the synergid.